of key area. This bit's really quite important because as you're going through the course, really at this stage, advanced hire is kind of this bridging point between school and university, and it is really good if you can start to develop some of the skills to extract the information yourself and learn it yourself from basically what should be being taught as we go along. So what I'm going to do is very, very quickly, hopefully in under 10 minutes, this is actually going to be the start of the lesson really. I'm going to show you how I'm going to plan what I'm going to do, what I'm going to cover, and how much I think I can get through. So I am aiming for our videos to not really be in excess of 50 minutes. I appreciate that this is the time it takes is probably a lot longer at home than it would be at school to get through some of these things. So we're not doing laboratory techniques for biologists. The reasons for that should be fairly obvious. Hopefully we will come back and do all of this stuff once we get back to school. So what we are starting on, and why your first lesson was based a little bit on chemistry, is proteins and proteomics. Proteomics is the study of proteins, usually the study of proteins on a fairly large scale. So how these proteins interact with other things, how their conformation changes, what role do they play, and how do they do their job. So I'm basically being told here that I've got to cover what the proteome is, and that I've got to go back and really cover the genome as well. So I've got the proteome and the genome, and I'm being told that the proteome is larger, produces a lot. The proteome has a larger set of proteins than the actual total number of genes. That's because we've got this alternate RNA splicing. There's also some pretty interesting things with somatic mutation in certain white blood cells and B cells to produce antibodies. I don't know if we go into that much detail in the course, but certainly alternate RNA splicing, and we've got some post-translational modification in there. So the first thing that I'm going to really list is that I need to take you guys through the proteome. I want to recap your gene to protein stuff from higher. That's going to be really helpful in understanding it. Now, post-translational modification was dropped from higher, so we need to actually do that separately. So we need to talk specifically about some post-translational modification. And then we need to talk about cells overall. So this little sentence here, not all genes are expressed as proteins in a particular cell. What I want to do is really talk there about differentiation and cellular tasks. And I want to go into what really usually using humans as the basis for this, talking about what the different proteins do, and also what cells in particular show those proteins and why, and the systems by which they might not produce proteins. So using highly dense chromatin structure, which really means we need to go back and recap histone proteins and chromatin. So we do need to go back and cover that a little bit as well. Then probably one of the most interesting bits is this conformational change, and this is where a lot of that chemistry will come in. So we will take a look at the sequence of amino acids, and this is your primary structure, and we'll need to get a note about that. So amino acid sequence determines protein structure, and then we want to talk about the primary structure. Then we want to go into this hydrogen bonding along the backbone results in the secondary structure, and I want to show you some alpha helixes and some beta pleated sheets. Now, this bit here, I want to show you the pi mole stuff, and there's this word isoelectric that has come up in the documents, so we need to cover that. We want to cover electrophoresis as well. So these words may not, you don't need to actually know these words because they're in the suggested activities, but because they're in the suggested activities, there may be problem-solving questions that are based around them, so it's a good idea to cover any new words that are there. We are going to cover chromatography in the lab skills part anyway. So, the structure of amino acids includes those R groups that we've spoken about before. Now, we need to add in some key words there, and we'll take some notes as to particular amino acids and show which ones are basic, acidic, just polar, and those ones that are hydrophobic, aka non-polar. Then the structure is going to fold, so those alpha helixes and beta pleated sheets are going to curve into an overall structure caused by the R groups. So we are going to need to cover what ionic bonds are. We're going to need to cover what van der Waals interactions are. And that will include hydrogen bonding. And we're also going to need to include some disulfide bridges. So what I want to do is to go through this bit first with you and highlight a few of the amino acids, pick out those amino acids and use them as a basis for discussion. I'm going to use the basis for that discussion will be some of the pi mole stuff, which is a computer modeling system. Then we are going to talk about those R groups and how we get those hydrophobic regions, ionic bonds, van der Waals interactions, along with the hydrogen bonds and the disulfide bridges. All the stuff from the first lesson will be heavily involved in this little bit here. Prosthetic groups. This is a non-protein unit that is added to finish off the functional protein, essentially, and that produces our quaternary structure, and that is basically the protein that can go on to do its job. So those prosthetic groups that are added on, they can sometimes be sugar molecules or something like iron, and we can talk about those in quite a lot of detail as well. So really, this lesson is probably going to be quite bulky, and it's going to include all of this stuff. So now I have to go away and turn this information into a lesson in terms of how I would go about this. Interestingly, this little bit here and this little bit here, I have found some articles that we will go through together, hopefully, as well. So I will be putting those up on Teams, and that'll be a place where we can move through it, and hopefully there's lots of questions from it. I'm hopefully going to show you how to start reading these and how to start turning an exercise of just reading into an exercise of learning quite a lot of information from it, because you shouldn't know all the stuff that's there, certainly not in a scientifically published peer-reviewed article. I don't expect you to know all of the stuff. I don't expect myself to know all of the stuff. A lot of reading those is about finding out what you do and don't know, and how to go about learning those things that you need to know. So that's what we're going to cover. These are the learning intentions, and hopefully that was a little bit insightful into how you can go about breaking down what is a fairly unattractive looking document into something that you can potentially take something from. Right, so that is the preamble essentially, and the learning intentions. What I'm going to do is turn this into a list of learning intentions, and we can get the lesson started. Okay, advanced hires, so this is going to be a very quick recap of the things that you should have covered in higher biology, and we're going to get up to the place where advanced hire really wants to take over and look a little bit deeper into our protein structure. So, going back to basics, our DNA strand is a double helix made up of several different bases: adenine, guanine, cytosine, and thymine. The backbone itself, the thick black line down here that's unbroken, that's held together by strong covalent bonds. That's very unlikely to break without enzymes getting involved. The strands themselves are held together by hydrogen bonding. That hydrogen bonding, if we remember back to our first little preamble into the chemistry for advanced hire, we've got a different attractive force for electrons depending on what element we have. So if we've got oxygen, that's actually got a fairly strong attractive force for electrons. It's 3.5 on the Pauling electronegativity scale, meaning that it's got a greater attractive force for electrons than the hydrogen. This leaves the hydrogen with a deficit in electrons, and so it is slightly positively charged. On the other side of the DNA strand, you may have something like an oxygen with a double bond to a carbon. That, because the oxygen has a greater pull for the electrons than the carbon, you're going to end up with a slightly negatively charged oxygen atom, because it's quite dense in electrons, because those electrons have shifted from the carbon, from that midpoint, more towards the oxygen. The fact that we have a slight negative charge here and a slight positive charge here creates an attractive force between these two atoms, and that produces that hydrogen bond that we see there. And that hydrogen bonding holds the two strands together. Now, when you looked at PCR, you found out that you can actually separate those strands of DNA at about 95 degrees. And if you think about it, the water molecules themselves like this have a slight negative charge here and a slight positive charge here, and there's a hydrogen bond between these two, very similar to this, and water will break apart and form steam or gaseous water at 100 degrees. At around that same point, you're going to be able to separate these strands of DNA. Now that we've covered that, we take a look at DNA in a slightly more living manner. So we've got, really, our DNA doesn't just hang about like this. It's going to wrap itself around histone proteins, and that DNA, along with histone proteins, is going to form our chromatin structure, which is really what our chromosomes are. that chromatin structure can either be dense or loose. Now, if you've got dense chromatin, the cell's unlikely to be transcribing and translating information from this bit very often. It's probably not going to be using the genes that are wrapped up in this section a lot. However, where it's loose, it's got easy access to those genes. It's probably going to be transcribing and translating the information in this section more often. So this is one way cells can differentiate themselves and specialize in particular protein production, is by having some areas with high density chromatin and some areas with low density or loose chromatin structure. So from our section that codes for a protein, we really describe that as a gene. The word gene can sometimes get a little bit hazy. However, as far as the definition we want to use will go, it's a section of DNA that will go on to produce a functional protein for our cell. So that gene is transcribed into RNA. That RNA is single-stranded, and that RNA will contain both introns and exons. The RNA splicing involves removing those introns and splicing the exons together. Now, what could happen is you could have alternate RNA splicing where different exons are selected, and so that one gene could go on to produce several different proteins, and that's why the proteome, it's one reason why the proteome is much, much larger than the number of individual genes found in the total genome. Once we have got our mature mRNA, then we go into translation and we will produce our protein, and that is really where we want to begin today, is looking at the individual amino acids and how they interact with one another to form a protein. Okay, advanced sciers, so we've just covered the stuff you should have picked up at higher, which is really the transformation of the information in the DNA through transcription to RNA, and then finally translation of that RNA code into a protein, which is really the specific sequence of amino acids to produce a functional protein for the body. Now, this is where the advanced hire kicks off, and the title for this bit should be Proteomics, and that is the study of how proteins work, essentially. And the proteome is really the total number of proteins that any organism can produce. Now, that proteome is actually a lot larger than the genome itself. So what we're trying to say there is the total number of proteins that can be produced by the genome is a lot larger than the actual number of genes that we find in the genome. That is due to the alternate RNA splicing and something called somatic recombination that we get in some types of white blood cells called B cells and certain types of T cells. The proteome of any organism will be more similar the more genetically related they are. So your proteins are going to be pretty similar if you're pretty closely related to another organism. Now, what we're going to take a look at is how the primary structure, aka just the sequence of amino acids that we find through certain types of bonding, allow the protein to take up a very specific shape. Now, that the first thing that we'll talk about when we're talking about those specific shapes is the secondary structure, and that is the formation of alpha helixes of within the protein and the parallel and anti-parallel beta sheets. We're actually going to use PyMol to look at that a little bit later on. So we're going to have a little bit of a look at a computer program that allows us to see and model these amino acid chains as they would appear when they're folded up as a protein. So this is really just the first little step. We'll start to move on to the bonding that allows this secondary structure to take place and the bonding that allows the tertiary structure to take place. So we'll get both of those down. Okay, so this is really just our note for the R group and the functional bits that we'll find there. So our basic amino acids, which is a bit weird to say, but they will create a basic environment when they are placed into it. They have a positive charge. Acidic amino acids have a negative charge. Some of those R groups will be hydrophilic because they're polar. The example that we used with alanine was actually a non-polar R group that would be hydrophobic. These things here, along with some of the other types of bonding that we're going to speak about later on, help to form the tertiary structure. So we get these major parts that become alpha helixes and beta pleated sheets, and that then folds into a three-dimensional shape for our polypeptide. So that's just the note for the R groups and the functionality that they bring to the table. Okay, so before we move on to talking about how our chain of amino acids, given the primary structure of our protein, will fold into a specific shape, we've really got to talk about the types of bonding that can be produced because of the R groups that we've got. Now, the amino group and the carboxyl group that is present in all amino acids, I'm going to do it in this colour. It is a good idea every time you get the opportunity to try and draw out an amino acid. It isn't a requirement that you remember what all the amino acids look like, but the more you do it, the more familiar you'll become with them. So I'm going to start with my central carbon and put a hydrogen there. I'm going to put my carboxyl functional group here, and I'm going to put my amino group here. And that's how you know it's an amino acid. An amino group here and an acid group here. Now, the R group I'm going to do in black coming up here, and what we've got is 2 carbons bonded to hydrogens, and interestingly we've got another carboxyl group. Now this carboxyl group actually has the hydrogen has come off and has left a negatively charged oxygen there. The H positive is what makes the environment around this amino acid acidic. So that H positive is going to create an acidic environment, and this is an acidic amino acid. And acidic amino acids have a negative charge to them. Now, that negative charge will be attracted to positive charges, and what we're going to do is look at another amino acid that's actually basic, which has a positive charge. So what I'd like to show you just very, very quickly is this is what we have got here. So this is our carboxyl functional group, and this is our amino functional group, and then we've got our R group. Now our R group has 2 carbons with hydrogens bonded. This little section here will be essentially hydrophobic. We'll get onto van der Waals forces, but this bit really is going to increase the ability of this negatively charged section to reach over a distance. If this was shorter, then there wouldn't be as much movement in that R group. So this really allows quite a lot of rotation and movement for this negative charge to form a bond with something, and we'll talk a little bit about those ionic bonds that they can form later on. This here is just showing that negative charge, so we will go on and speak a little bit about how this negative charge can form up with a positive charge. Okay, so once again, I'm gonna start off with my part of the amino acid that is the same regardless of what amino acid we're talking about, and that is the amino group and the carboxyl functional group. And the R group for this one, this is going to be lysine. Now we've actually got quite a long carbon chain here. The carbon chain's actually 4 carbons long. And this is all aliphatic, which just means it's carbons and hydrogens all the way up until we get to here. And then we get an N with 3 hydrogens attached. Now, nitrogen has a valency of 3. It can usually only form 3 bonds. If it's formed 4 bonds, then there will be a positive charge associated with this, because basically what's happened is, in fact I'll use that as a wee example, the lone pair of electrons there has allowed an H positive to bond. Because the H positive has moved out of solution, the solution itself will become less acidic and more alkali, and this is why this is a basic amino acid. So this is a basic amino acid and it has a positive charge there. Now, what we can do is, once again, we can take a little look at this one. You'll need to excuse the silver oxygens. I ran out of the red molly mods, so you'll have to just pretend those are red. So once again, we've got our amino group here and our carboxylic acid here, and this will help form the peptide bonds going in either direction to form our protein. Now the R group for this one, once again, pretty interesting that we've got this really long section of hydrocarbons, and this is going to allow this R group to have a lot of movement. And what we're going to do is we are going to take a very quick look at how these 2 amino acids, the glutamic acid and the lysine, can play off against each other and form an interesting little bond that will help determine the eventual shape of our protein. Okay, so for this example, what I'm going to do is I'm going to take glutamic acid, alanine and lysine, and we're going to bond them together in an extremely short peptide chain. You wouldn't get a peptide chain being formed that's this short normally. However, it's going to make the point and make it easy to compute for us. So what I'm going to do is I'm going to draw out glutamic acid, alanine and lysine, then we're going to bond them together, and then we're going to talk a little bit about what impact that will have. So what I'm going to do is I'm just going to have my alanine in the middle here, and I'm going to have lysine on this side, and the one here is going to be glutamic acid. Now, what you'll notice is I haven't actually put the R groups in yet. I'm going to truncate these right down. So all I'm going to do here is put my CH3, because that is what we've got on alanine. Now, the lysine, the R group here is a positive charge, and I'm just going to write a plus charge there. And the R group here is a negative charge. I'm just going to write that negative charge up there. Now, what we're going to do next is we are going to bond these together. We do that with our condensation reaction that will produce water. And so we'll get H2O coming out here and an H2O coming out here. And we're going to link that carbon with that nitrogen. And we're going to produce our polypeptide chain. So what we're going to have, hopefully if I don't mess anything up, is our carbon, little hydrogen there. And this will go on to a nitrogen, a little hydrogen there. Right, so what we spoke about just in the few clips before was that the R groups for both of these have actually got pretty long hydrocarbon chains that the really functional bit, the charged bit, is at the end of. So these are actually quite long. Now, what we would imagine to happen, potentially, is that negative charge there, and that positive charge there will be a force of attraction pulling those together. Now, this bit's non-polar, so it's almost going to bend away and out from that that positive and negative charge being pulled together. So we're going to have these amino acids moving round towards each other, and that's going to cause a change in shape. Now, what I'd like to do is show you that with mods just to make it a little bit more visual for you. But really, all we want to be talking about is that negative charge and that positive charge on the glutamic acid and the lysine pulling together, and between these two will actually form an ionic bond. We will get onto that a little bit later on, but that's what I want you to be visualizing. The fact that we've got these amino acids in a primary structure, aka this is the sequence that they come in, results in a very specific type of folding due to the R groups having different properties. In this case, a negative charge and a positive charge. Okay, so this is my 3 amino acids. We've got our amino group here, our carboxyl acid here, our amino group here, our carboxyl acid here, our amino group here, and our carboxyl acid here. Bearing in mind these should be red, and I ran out of the red ones. So what we're going to do is we're going to bond these together. That's actually going to liberate water, and I'm going to use that to fix this as we go. Don't let that confuse you. I'm just really wanting to highlight what's going to happen here. So we are going to have that hydrogen come off. Well, one of the 2 hydrogens come off, and we're going to form a water molecule. So that water molecule is going to leave. That is a condensation reaction, and we're going to get a bond forming between the C that has a double bond to that oxygen and that nitrogen there. That water just becomes another water molecule, and I'm going to use that oxygen just to fix this one so it looks correct once we get to the end. So ignore that bit. Then we're going to get another bond forming, and we're going to have the hydrogen coming off the nitrogen here, and the OH coming off the carboxyl functional group, and we're going to get water, liberated once again, and that's going to fly away, or swim away, however you want to think about it. And once again, I'm going to fix the fact that I don't have enough molly mods. And we've got our peptide bond forming there. So, now, we've got a pretty long polypeptide chain. This R group here is from our alanine. This is our lysine, and this is our glutamic acid. So, this molecule is able to, all of this is moving and rotating about, and just doing that, basically. Now, what we've got is this non-charged hydrophobic part here that's not really going to want to take part. However, there's a big old positive charge here and a big old negative charge here. What's going to happen, potentially, depending on what other interactions are taking place, is that these two are going to move round and form an ionic bond there. Now, you will note that that's a very different shape than that. That's it flat laid out. However, because we've got that negative charge there and that positive charge there, it's going to move round and it's going to take up a very specific shape. Now, that very specific shape is because of the primary structure, and that is the order of the amino acids. If we had ordered the amino acids differently, we would be getting a different shape. Now, depending on where these bonds and other types of bonds are that we'll come on to later, we will get the production of alpha helixes and parallel and anti-parallel beta pleated sheets. So this is really how I want you to be visualizing it. Just the R groups creating ionic bonds and the shape conforming like that. And we've got our strong covalent bonds holding our amino acids together. So we have just seen the ionic bonding that can form between glutamic acid and lysine, and that's because that glutamic acid has that formal negative charge and the lysine has that positive charge, and they will form that ionic bond because you've got those two fully charged groups at the end. That's going to be a relatively strong bond and pretty hard to break, and the polypeptide chain will fold to allow those type of bonds to take place. But they're not the only type of bonds that help form our secondary and tertiary structures. So what we've got to talk about is the van der Waals forces, and these are really split into two major groups: the overall electron displacement and the random movement of electrons. So if the electrons are displaced overall, you've either got hydrogen bonding or a dipole to dipole interaction. Now, a hydrogen bond and a dipole to dipole is really created from the same thing. However, the hydrogen bonding simply denotes a little bit of extra strength. So in the example here, we've got water. Water's electronegativity is 3.5 on the Pauling scale, and hydrogen is just 2.2. So the electrons find themselves well and truly pulled towards the oxygen, making that a fairly strong, not a full formal negative charge, but a fairly strong negative charge on the oxygen and a fairly strong positive charge on the hydrogen. Whereas with the dipole to dipole interaction, what you'll have is just a very slight difference in the electronegativity, meaning that the electrons are only slightly pulled to one of the atoms, and that is going to create a very, very slight bonding force between the next molecule here. So you could imagine that there's actually a bromine here with a very slight positive charge. The dipole to dipole interaction would be between that chlorine and the next bromine up here. And that would be very, very slight because there's only a very slight difference in the electronegativity, and that would be a dipole to dipole. Hydrogen bonding is the same thing, it's just those electrons being pulled closer towards one atom, but it forms a much stronger bond. Now, interestingly, we've also got this random movement of electrons, and this is the London dispersion force, and that's the weakest of our forces. Now, we can describe that in a fairly simple manner. Okay, so to explain this London dispersion force, I'm going to use two methane molecules, and there's no dipole within this molecule. It's completely, as far as it can ever be, it's non-polar. So the only force of attraction between these two molecules can be the London dispersion force. Now, the London dispersion force is created from the random movement of electrons. So what we might find at one timestamp, one split second, is that the electrons on this molecule happen to be clustered over at this side here. Now that's going to create a very slight negative charge, and as you know, that slight negative charge is going to push other negative charges away, and so on this molecule we might see these electrons being pushed to the other side of the molecule. Now this has created a situation where there is a very, very, very slight positive charge on this side of the methane and a slight negative charge on this other side of the methane. This is thousands of times less than you would find in a dipole. So this is much, much weaker, and it really is very, very weak. But there is still some attractive force between these two things here. So you've got to remember that London dispersion forces are part of how our proteins take up their shape, and they will influence things very slightly. When we take a look at the PyMol computer program, we'll try and speak a little bit about how that might take place. We're going to have to do a little bit of imagining as well. Is admittedly visualizing how all of these bonds create the conformations in proteins is tricky, and it will take you a bit of time. Now, this is actually so weak that between these two methane molecules, you would actually have to reduce the temperature to minus 162 degrees Celsius to actually get enough of a bond here to actually form a liquid. So anything above minus 162 degrees, the vibrations of the molecule would be enough to separate them into a gaseous state, whereas at minus 162, there's so little vibration in the molecules that this very, very weak London dispersion force can hold them in a liquid state. So that is your London dispersion force. Okay, so the last type of bond that we've got to talk about is the disulfide bridge. Now, once again, this is actually just a regular old covalent bond, but because it's very specific to proteins and it's really important that some proteins form these, it's given that specific name. So we're going to use cysteine as our example here, and that's an amino acid that has a sulfur atom in its R group. So the first thing that we've got to do in this little example that we'll do is form our peptide bond between the two amino acids by removing our water molecule. So that's just what we've covered before. Now, once that peptide bond has formed, we are going to have this sulfur and hydrogen sticking out of our R group. Now, you don't need to know how the cell does this. It's part of the processing of the polypeptide tertiary structure into its final quaternary structure, or even just from the primary structure into the secondary structure, or for the secondary structure into the tertiary structure. It is part of the process of making the protein functional and to making sure its shape is correct for its particular function. So this process is called oxidative folding, and it's catalyzed by enzymes. Now, where we had this sulfur and hydrogen up here, we're going to remove the hydrogen and we're going to form a covalent bond between those two sulfur atoms. That may cause, in this example, may cause the amino acids to pull up slightly. However, this bond could be formed between amino acids hundreds of amino acids apart in the actual protein. So we will take a little bit of a more detailed look at that when we take a look at the PyMol program later on. But that is our disulfide bridge. Di meaning two, sulfide meaning sulfur, and we've got two sulfurs holding in a covalent bond holding these two amino acids together, not just with the peptide chain but with that disulfide bridge, which will cause a conformation change. Now bear in mind that they don't need to be adjacent. They could be hundreds of amino acids apart, and the amino acids could have folded right round and a disulfide bridge forming between the two, creating a loop in the protein. But we will, like I say, take a look at that with the PyMol app. Admittedly, there was quite a lot that we covered there. There's a fair chunk of chemistry that we really recapped and went back over again. We spoke about the R groups and those R groups having features that allow specific types of bonding. The main things that we really want to take away from today is that primary structure of our protein is the sequence of amino acids. So that is simply the order in which we find the amino acids, and that order will create a very specific shape because of the bonding. Now, the first major conformational changes we talk about as the secondary structure. So these are like the big main things that form, and these are the alpha helixes, the parallel and anti-parallel beta sheets. So these form from the fact that we've got this specific sequence, and we'll see these in the next lesson fully, and we'll be able to appreciate what these look like, and we'll be able to talk about why they come about. Now, because of all the bonding we get the tertiary structure. And this is very simply just the three-dimensional shape that the polypeptide takes. It might be that this tertiary structure is the full protein, but it might not be. And what we'll do next time is we will talk a little bit, we'll go through this all again, and we'll add in the quaternary structure, and we'll talk a little bit about post-translational modification. But I think that is more than enough for you to wrap your heads around today. I will be posting up on Teams a scientific article that we are going to read through together, so I'll post that up on Teams today as well. And so you've got this lesson and the lesson before to go through, and we'll take a look at that scientific article. I'm hoping that I can get up to pace, and that we can get at least 2 or 3 lessons done a week on YouTube, and the scientific articles and some other work will take up the rest of the time. But admittedly, it's taken a little bit longer to do the videos than it would be to do a normal lesson. So hopefully this week you'll get another full 40 to 60 minute lesson on Wednesday, and we will take a look at that scientific article as well, and we will take a look at that PyMole program as well, which I think will really help bring it all together.